Well, hey, it's Thursday, Living Environment fans. Less than a week till the Regents exam day. Yes. As you may notice, I'm not in the office today. I'm attending to some other business, so in my stead, there's a substitute who I don't know who that is, but they will, of course, lead you through today's program, which includes this Bellringer, Ed Puzzle video, and then more Regents practice. Okay, so I want you to do the Regents practice. And then at the end of the period, myself will share with you the answers to the Regents practice. Hopefully that makes sense, right? Just like I would normally do with you. All right, so that's today's focus. Regents review, Regents practice, Regents, Bellringer. All right. Hey now. Mr. Kovacs class. It's Mr. Kovacs class. You might learn a ton. It's Mr. Kovacs class.
He's interesting too.
It's Mr. Kovacs class.
And he's super cool.
It's Mr. Kovacs class. Does like to ask about gene technology, like what you've learned about various technology with genetic engineering and that kind of thing. All right, and so just so you know, like gene technology is nothing super new, but it's used a lot to manipulate the genes inside of cells. And some things you might be asked is like, well, here, let me give you some examples. Might throw in something about recombinant DNA. Recombinant DNA is when you effectively cut out one portion of a gene and you stick it on to another portion of a gene, recombining DNA from two different organisms. Okay, to do that, you need what's called a restriction enzyme. Restriction enzymes, they cut DNA in specific places so that then they can be stuck together, right? That's how you can take genes from one species, cut them out, and then move them into the genome of another species. And we've talked about some examples of this for sure. This is how insulin is made, right? Like insulin is a protein, that's a hormone protein that basically is the key to unlock the door to let glucose into muscle cells or into any cells, really. Okay? You make it in your pancreas normally. If you have diabetes, you don't make insulin. So what do? We use recombinant DNA technology where we clip the gene for human insulin and we insert it into the chromosome or the genome of bacterial cells. They can read it, so therefore they make insulin. They don't need the insulin, so you can capture it and bottle it and sell it to somebody with diabetes and save their life, right? Recombinant DNA technology, gene and genetic engineering are somewhat synonymous with each other. Remember Andy the monkey? Another example of, he's a transgenic animal. So yeah, they use recombinant DNA in a multicellular organism to make Andy. And hopefully, do you remember what Andy does that most monkeys don't? That's right, he glows, right? He glows because they were able to move a jellyfish gene, a protein made by jellyfish that glows in the dark, so to speak, and move it into his genome. And not only that, not only could he read the DNA, right, the jellyfish DNA, but because he glowed in multiple places, that means he could copy the jellyfish DNA and pass that copy along to other cells. And that jellyfish DNA could be read by all those cells, by the ribosomes of those cells, because we see the proteins. Which means if you take the next step, he could potentially copy that gene into his gametes and pass it on to the next generation of monkeys. And you could have the beginning of a brand new adaptation for these monkeys of glow in the dark, which might be a benefit. Maybe it's a benefit for attracting mates or something. Maybe it's a detriment for playing hide and seek. I don't know. When we talk about genetically transgenic organisms, when we talk about food products, we tend to call them GMOs, genetically modified organisms. And GMOs, to be honest, usually has kind of a negative connotation, but GMOs are not necessarily good nor bad. They are just modifying the genes found in crops to make them better able to survive, to make it to market. So GMOs are designed to increase food productivity. So to me, that seems like a good thing. but it is tinkering with genes, which some people don't like. So might be asked in the region some ethical questions about tinkering with genes with various organisms. Of course, there's cloning. Cloning is genetic engineering, where you take the entire genome of one organism and you basically replicate it into a different organism so that they have the same exact genes. Okay, this is kind of what bacteria do all the time when they copy and split through mitosis, but it's abnormal for multicellular things. And Dolly, of course, was the first successful cloned large animal, and she happened to be a sheep. Dolly, they cloned Dolly by what's called nuclear transfer. So they took a cell from a living sheep, took out its nucleus, then put the nucleus into the egg cell from a female sheep. They did like a nucleus swap. Right? Now you have a full nucleus. You implanted that nucleus into the uterus of a surrogate mother, and she gave birth to the clone of the original donor sheep. Right? Cloning. And there are lots of advantages, I said, to using cloning technology because you guarantee what you're going to get. Usually in sexual reproductive organisms, you don't know what you're going to get, but now you know. It could be really advantageous, especially if you're into like livestock and growing animals for food or for clothing or those kinds of things. Don't you think? And I think I also told you the story of Casey, the cow, who is wild because he's a clone of another cow that had already died. And that leads us to a whole new level of probabilities, of possibilities. You could find DNA from things that are dead. If you could find a surrogate mother to carry their genes, you could potentially give birth to animals that are extinct, right? Like mammoths. The hard part would be finding a surrogate mother. You'd have to find an animal as big as a mammoth who could carry that mammoth baby. Or how about endangered species like Siberian tigers, which are going extinct. There are not very many of them. You can help them reproduce by just cloning them, getting them back in there. And then my favorite thought is that if it works for big animals like mammoths and tigers and sheep, it would work for us. How cool would it be to clone yourself? I mean, think about it. If maybe you're in a relationship where you can't have children because of some medical problem, you could clone yourself. You just need a surrogate mother, clone yourself, and now you have a genetic copy of you, a genetic relative of you. Or if we could bring back, the dead. I mean, Babe Ruth. I always say, if you could clone Babe Ruth, teach him how to play baseball, he'd be the best to ever play.